Non-canonical base pairing, an interesting facet that has contributed to the flexibility as well as the functionality of nucleic acids. Now, what do we really understand by non-canonical? When you have a base pairing, and if the base pairing is deviating from a conventional rule or from a conventional system, then that is what is understood to be non-canonical. Well, this is with respect to base pairing, but this could non-canonical could be respect to any basic system. Say, for example, translation. You have a conventional way of translation occurring, but suppose it deviates from that conventional rule or the system, then it is termed as non-canonical. So therefore, the session is going to focus on various non-canonical base pairing that occurs in nucleic acids, both DNA as well as RNA. So let us look at the learning outcomes of this session. One always relates to the standard Watson and Crick base pairing. So that is what is conventional. That is what is a standard. Okay. However, interestingly, there are many non-Watson and Crick base pairing observed, and this has immensely contributed to the different structures within the helix. Non-canonical base pairing is observed most commonly in secondary structures of RNA and are observed to be less stable than Watson and Crick base pairing. However, they have been observed. There are majorly two types of non-canonical base pairing, Hoogstein base pairing and Wobble base pairing. And in DNA, these can exist in equilibrium with the Watson and Crick base pairing. X-ray crystallography and computational studies have revealed various possibilities of non-canonical base pairing. So let us just look at and understand what we mean by Watson and Crick base pairing. So in a double-stranded DNA, which is a normal helix, we know that the helix is stabilized because of the hydrogen bonding between the nucleobases and also because of the hydrophobic interactions. Because we all know that individual bases are not soluble in water, and therefore they tend to move away from water and interact with each other. So therefore, that hydrophobic and also base stacking. So all these contribute to the double helix becoming stable. And we also know that depending on the way the normal Watson and Crick base pairing takes place, you have what is called as the major groove and the minor groove. And this is being contributed by the canonical base pairing itself. So the understanding is that if you do not have canonical base pairing, will there be a major groove and minor groove? There may be a major groove and minor groove similar to that of the Watson and Crick base pairing, or it may differ. Now to look at the way the Watson and Crick base pairing stands is, say you have the guanine over here which interacts with the cytosine, and it is always the 1, 2, 6 elements of the nitrogen guanine, of the purine guanine, that would interact with the second, third and the fourth atoms of the pyrimidine. So this is the normal or the conventional Watson and Crick base pairing. Similarly, you will find that in pyrimidine again you have the 1, 2, 6 phase of the adenine that is interacting or hydrogen bonding with the 2, 3, 4 phase of the thymine. So these are the conventional Watson and Crick base pairing that was proposed by Watson and Crick themselves, and these are what falls under what we call as a standard Watson and Crick base pairing. Now, it is interesting to note that the ribose or the sugar of the deoxyribose of the Watson and Crick base pairs are on the same side. If you draw a axis of symmetry this way, then you will find that the R in both the AT and the GC base pairs are on the same side. So this is also something that is observed with the Watson and Crick base pairing, standard Watson and Crick base pairing. Now let us look at therefore the different non-canonical base pairings that have been observed in nucleic acids. So Sanger in 1984 proposed that if you have base pairing between two nucleobases and the number of hydrogen bonds is 2, then you can have a non-canonical base pairing. So ideally what is suggested is that between two nucleobases, if you have two hydrogen bonds, you can have non-canonical base pairing. Now, looking at therefore these attributes and through various X-ray crystallography studies, what has been observed is the base pairing therefore can be classified into 12 families. And interestingly, what has been observed is that in these 12 families, there is a property which is called as isostericity. We would be looking at what isostericity is. But this isostericity is, say, for example, observed in all RNA secondary structures. Okay. Now, the RNA secondary structure can be formed when one RNA itself turns over each other to form the stem and the loop structure. This is one way of secondary structure formation. Or you can have two single-stranded RNA coming together to form hydrogen bonding between each other, and therefore this is also a secondary structure. So these can have what is called as the isostericity. Now, half of the families, okay, would have the ribose, if you consider the RNA, in the cis orientation, okay, and half of the families will have it in the trans orientation. So we'll just look at what do we understand by cis orientation and trans orientation. But interestingly, what is observed is that classified into the 12 families, one of the family is the canonical Watson and Crick base pairing. Okay, and this canonical Watson and Crick base pairing exhibits the cis orientation, that is where the sugars are present in cis orientation. All the other 11 families, they actually contribute to and has been observed mainly associated with secondary and tertiary structures of RNA. And many a times these secondary and tertiary structures of RNA form what is called as RNA modules. And it is these RNA modules, especially in the non-coding RNA. So when you say non-coding RNA, you can say, for example, think of tRNA. You can say, for example, think of introns. You can say, for example, think of small nuclear RNA or ribosomal RNA, or you can also think of siRNA, miRNA. These are all non-coding RNA. So you will find that such RNA modules have been formed in the RNA structure due to the non-canonical Watson and Crick base pairing. And all of these are basically having specific biological functions. So the RNA modules that are formed because of non-canonical base pairing lead to specific biological functions. Now, we told that you have basically, when you're looking at a canonical way, Watson and Crick base pairing, you have the sugars in the cis orientation. What does that mean? So when you have the base pair and you draw the line of symmetry or an axis of symmetry in this direction, okay, you will find that the sugars of both the nucleobases are on the same side. So this is what is called as the cis orientation. But when you have the axis of symmetry drawn between the same nucleobases, but with one sugar being one side of the axis of symmetry and the other sugar being on the other side of the axis of symmetry, then this is what is considered to be trans orientation. So you may either have the sugars as cis orientation or you may have the sugars as the trans orientation possible in a non-canonical base pair. Now, therefore, depending on the way the base pairs associate with each other, as mentioned, there are 12 base pair families. So here you can see Watson and Crick, Watson and Crick. So you can see the base pairing is Watson and Crick, and this is the cis orientation. So this is what is the canonical Watson and base pair, Watson and Crick base pairing. But you may also have the Watson and Crick base pairing, but the sugars not being in the cis form, the sugars being in the trans form. In such a situation, it is not considered to be canonical. Again, it becomes non-canonical. Likewise, you can have Watson and Crick, and S, this S stands for the sugar edge, this H stands for the sugar edge. So basically we are looking at different interacting edges of the nucleotides that can form hydrogen bonding, and based on how they interact you have 12 different basic families. So we'll look at what are these interacting edges. So say for example you consider a purine nucleotide. So in a purine nucleotide, as you can see, this is the ribose, okay? This is the phosphate that is associated with the 5' carbon of the ribose, and at the 1' carbon you have it associated with the N of the purine. So you always have N9 of the purine associated with the C1' of the sugar. So you know that the N-glycosidic linkage in a purine is always between the first carbon of the sugar and the ninth N of the purine residue. So therefore, when you have the nucleotide placed like this, we obviously, when you look at the numbering that is there in the nucleotide, it is always 1, 2, 3, oh sorry, 1, 2, 3, 4, 5, 6. So this ring covers the 6 skeleton of the 6 atoms of the skeleton, and then you have the 7, 8, 9. So this is the 7, 8, 9 phase of the purine molecule. Now, whenever there is any hydrogen bonding with the 6, 1 and the 2, okay? So you have 1, 2, 3, 4, 5, 6. So when you have 1, 2 and the 6 interacting or hydrogen bonding, then this is what is called as the Watson and Crick edge, because it's found that in the canonical, or the one that was proposed by Watson and Crick, it was these that formed the hydrogen bonding. And so therefore this forms what is called as the Watson and Crick edge. Suppose you have the atoms present over here that forms hydrogen bonding, then it is what is called as the sugar edge. And suppose it is basically the 7, 6, 9, or 9 is not possible because it is already in a linkage, but suppose it is 7 and the 6, okay, this edge that is forming the hydrogen bond, then that is what is called as the Hoogsteen edge. The reason why this is called as Hoogsteen edge, because the first time that a non-canonical base pair was identified by a scientist called Hoogsteen, and he showed that the hydrogen bonding was not with the 6, 1 and the 2, but it was more with the 7 and the 9, this phase, and therefore you have this edge called as the Hoogsteen edge. Similarly, when you look at the pyrimidine, okay, again you know that the glycosidic bond that is formed between the sugar of the ribose, sugar of the RNA with the sugar of the ribonucleotide with the nitrogen base, it is the one nitrogen of the pyrimidine that is forming the glycosidic linkage with the first carbon of the ribose. So it is basically a C1 prime 1N glycosidic linkage, which means that in the pyrimidine you would always again find that the numbering is 1, 2, 3, 4, 5, 6. Okay. So in such a situation, what you observe is that the 2, 3 and the 4, okay, these portions are the region that form the hydrogen bonding with the opposite nucleobase that is present over here. And so this is what is called as the Watson and Crick edge. Suppose you have hydrogen bonding formed by this and this, then this is what is called as a CH edge, but to make it simple, many people call it as a Hoogsteen edge even for pyrimidines. Similarly, if you have this portion only interacting to form the hydrogen bond, then this is what is called as the sugar edge. So what we observe very clearly is that each nucleobase has 3 interacting edges, and therefore there is a possibility of forming non-canonical base pairs. It's not just that there is only Watson and Crick edge, but there is a sugar edge and a Hoogsteen edge as well. And so you can have hydrogen bonding in different ways happening between two bases. Okay. So therefore, this is an important facet about the non-canonical base pairing. Now let us look at isostericity. So when you look at the structure, you can already see that the G and the C is a canonical Watson and Crick model where you have the 2, 1, 6 and the 2, 3, 4 of the pyrimidine interacting. So this is the Watson and Crick edge of the purine interacting with the Watson and Crick edge of the pyrimidine. Okay. Plus, you can also observe that the sugars are the cis form, because if you draw axis of symmetry in this way, then you will find the sugars are present on the same side of the axis, and therefore it is the cis form. Now when you look at the A and the U, again A and U is canonical. You can see that the Watson and Crick edge of A is interacting with the Watson and Crick edge of the U. But what is isostericity? Isostericity is when the distance between the C1 prime carbon of the sugar, so this is the sugar moiety, so C1 prime carbon of this and the C1 prime carbon of the adjacent base pair is about 10.5 angstroms, then that is considered to be isosteric. And in a canonical Watson and Crick base pairing, so this is canonical, this isostericity is possible when it is the cis Watson and Crick base pairing. So this is a phenomenon that has been associated with the double helix, okay, where you have the sugar arrangements in such a way that they are 10.5 angstroms away, and if they are 10.5 angstroms away from each other and in the cis configuration, then or in the cis conformation, then that is what is understood to be isosteric. So two base pairs are isosteric to each other when they are 10.5 angstroms away and in the cis configuration. So the positions and the distances between the C1 prime carbon atoms are very similar. So if that is the case, then you have the isostericity. Now say for example, let us consider the same base pair, okay, so the GC and the AU. But now what you can observe over here is that the GC has the sugar in the trans form. So you can see this is the sugar over here and this is the sugar over here. So when you draw a line of symmetry, you have the sugars in the trans form. Similarly, in the case of AU, you have the sugars in the trans form. In such a case, if you were to measure the distance between the two sugars, the other sugar from this should have been at this position at 10.5 because this is 10.5. However, what in reality is observed is that this sugar over here is far away and not, you know, conventional 10.5 angstroms. So therefore, you will consider that this base pairs, the two base pairs are not isosteric. They do not show isostericity. So the complementary Watson and Crick, now we call it complementary because it is GC base pair, right? We call it complementary because it is AU base pair. That is what Watson and Crick had suggested, that it is always A interacting with U or A interacting with T and G interacting with C. So here when you have the complementary Watson and Crick base pairing, but the difference is that the sugars are no longer in the cis arrangement, they are in the trans arrangement, and just that one difference where the sugar is in the trans arrangement makes it non-isosteric. So it is no longer having the same configuration as a canonical Watson and Crick base pairing. Now it becomes non-canonical. Also, you can observe over here, the GC base pairing is happening, but because of the positioning of the sugar in the trans form, you do not have 3 hydrogen bonds between G and C, but you only have 2 hydrogen bonds between G and C. Say, therefore, again, this will be considered non-canonical. Okay? But interestingly, what has been observed is that non-canonical base pairs can have isostericity if they are changing their tautomeric form. So all of us know that the bases are existing typically in one tautomeric form. When they change the tautomeric form, then it is possible that you have the possibility of changing one tautomeric form into another tautomeric form. So basically, a tautomeric change of the C to its iminoform or the G to its enol form would be required for forming a trans GC base pair. Okay, so this GC base pair in the trans form also has been formed because the tautomeric form of one of the nucleotides or one of the nucleobases has changed. So that is something that is interesting. We will take it up just in a few minutes. But before we consider the transform or the form in which you can have isostericity even with non-Watson and Crick base pairing. See here, what we have observed is this is a non-Watson and Crick base pairing, but there is no isostericity in this case. So, say for example, you have the GU, this you know is non-Watson and Crick base pairing, so it is non-canonical. You have the AC, again this is non-canonical, but when both these base pairs are in the trans form, okay, then the distance between the two sugars, C1 position of the two sugars are equivalent or similar and are comprehending to 10.5 angstroms. So when you have something like this, then you can understand that even a non-Watson and Crick base pair is able to show isostericity. And this is more observed in helices that are formed by the two strands being parallel, not anti-parallel. We all know that in general, when you consider DNA, you have the two strands in the anti-parallel form, right? But in such cases where you have isostericity and you have transform, then it has been observed that the two strands that are coming together to form a secondary structure or to form a base pair, they are more in the parallel orientations. Okay, so these are certain observations and these are certain interesting observations because you kind of understand that a lot happens within the DNA or within the secondary structures that are formed in nucleic acids. So to form the isosteric conformations, what is important is the sugar conformation, whether it is a 3' endopucker structure or whether it is a 2' endopucker structure. The spatial arrangement of the nucleotide, especially the purine nucleotide with respect to the base. Okay, whether it is in the anti conformation or whether it is in the syn conformation. Also, whether you have the possibility of the trans or the cis arrangement of the sugars. So all of these factors will contribute to whether or not you can have an isosteric conformation. And this is exactly what was also shown by Richard Dickerson. Different forms can have different endopucker sugars. So you may have a symmetry or you may not have a symmetry based on whether it is a different endopucker sugar or it is either in the anti or in the syn form. So these things or these changes in the sugar moiety itself is affecting how you have within a non-canonical base pair, whether you have isostericity or you do not have isostericity. Now going forward, let us look at tautomerism again in a little detail. So whenever you have a chemical compound, say for example you have phenol, then phenol can exist as phenol or it can exist in the keto form. So the OH of the phenol can exist the way it is in the enol form, or when it becomes a double bond O, so when it becomes a phenone, okay, then it is a different form of the same compound. You see, the compound is not changing. The only thing that is changing is the distribution of protons and electrons within the structure, and therefore you have two different forms. One is an enol form and one is the keto form. So, most single chemical compounds that can exist in two or more interconvertible structures, okay, and they differ basically in relative position of one atomic nucleus, which is generally the hydrogen. So, we are talking about the protons basically. Then that is what is called as tautomerism. And the two forms that are there will always be called as tautomers, okay, and these type of isomer compounds, they only differ, as I mentioned, in the number of electrons and the protons, and they exist in dynamic equilibrium. So they can sometimes be present as enol form, sometimes be present as keto form, sometimes can be present as amino form, can be present as imino form. So all these are tautomers. And when you look at nucleobases, you will find that they too exhibit what is called as tautomerism. So you can have the adenine existing in the amino form. So when you have the NH2 present over here, or you can have the imino form. So what has happened in this is this proton or this hydrogen atom has moved from the sixth carbon, the sixth, the group on the sixth carbon to the first N. Okay, so you can just see that there is a rearrangement of how the hydrogen is present in the two ring structures. So therefore this becomes the amino form and this becomes the imino form. So basically we are saying that the adenine can exist either in the amino form or it can exist in the imino form, and they can be present uh, you know um, as in a dynamic equilibrium. But definitely what is noted is that most nucleobases try to typically be present as one type of tautomerism or one form or one tautomeric form. They are more stable that way. Okay. Now, if you see guanine, again, what you can observe is that this is the keto form where you have the double bond O, but you can also have the H from here going here. So it becomes the enol form. Okay. And because by lieu of the positioning of the H to the O, you can see that the double bond position has also changed, right? So you can have such changes in the positions or the number of electrons and protons, and therefore you have these two tautomers. Same you can see in cytosine. It can be present as the amino form or it can be present as the imino form. Or you can see in the thymine, you have the keto form and then you can also have the enol form. So you can also have something called the lactam and the lactim form as well. So these are basically tautomerics, that is only differing in the position of the protons and the electrons. And these tautomeric forms actually contribute to whether a non-Watson and Crick or a non-canonical base pair can be isosteric or not. So when you have modifications in bases, right? So if you have a nucleobase and if at one of the positions you have a base getting modified, so addition of a methyl group or an addition of sulfate moiety, things like that, if that happens, then it can alter protonation, and if it alters protonation, we can know that it alters tautomerism, and also it can alter the syn and anti-conformation. And all of this changes, whether there is a change in protonation or there's a change in tautomerism or there's a change in syn anti-conformation, they're going to affect the base stacking. And when they affect base stacking, you will find that the overall stability of the entire helix is getting affected. Even if it is a mini helix, it can get affected. So therefore, when you say mini helix, it means just a few base pairs coming together to form a secondary structure. Okay, then that forms a very small helix. But base modifications have been observed to affect the Hoogsteen edge, the Watson and Crick edge, and also the sugar edge. So if you have modifications at these positions, the hydrogen bonding would be affected with the opposite nucleobase. So this is something that is observed both in purines as well as in pyrimidines. So for example, over here, if the oxygen is substituted by sulfur, then it would definitely affect the Watson and Crick edge. Why? Because a sulfur will not be able to form a hydrogen bond. So that way you will see that many base modifications affect the structure or the hydrogen bonding, and that in turn can affect the base stacking, and therefore most DNA repair mechanisms present in the cell would try to correct the alteration that has happened. Now, when you look at again the same aspect with respect to, since we talked about tautomerism, you can see that here this is a normal standard Watson and Crick showing isostericity. So you can see that you have the sugars in the cis form, okay, you have the Watson and Crick edge of both the nucleobases forming the hydrogen bonding. Similarly here between A and U. But say, for example, you have non-Watson and Crick isostericity, then that Watson and Crick, non-Watson and Crick isostericity is possible if there is an alteration in tautomerism. So, in an A and C is non-canonical. But if the C or the A, either of this is existing in a different tautomeric form, okay, then you can have isostericity possible. So if suppose this is in the amino form and this is also in the amino form, then you will not have isostericity. But since the amino form of cytosine got converted to amino form, okay, you could have isostericity. You see the same over here. If this A would have been present in the amino form itself, then the amino of 2 would not have formed an isosteric configuration. But because one of them has changed its isomerism, you can see that it is existing in its isosteric form. So same goes with UNG. UNG, as you can see, is a Wobble's base pair, and you can observe very clearly that when U is in the enol form or when G is in the enol form, you will have isostericity even if it is a non-canonical base pairing. Now, you can see that when you have a canonical Watson and Crick, this part contributes to the major groove formation and this part forms a minor groove. And when you have isostericity between the non-canonical base pairs, you can see that they also contribute similarly to the major groove and the minor groove formation. So effectively what we are looking at is that if the non-Watson and Crick base pairs show isostericity, then they would have very similar minor and major groove formations. So the environments of the major and the minor grooves of the standard Watson and Crick base pair are the same in both the isosteric non-Watson and Crick base pair as well. Interestingly, what is observed is you can have non-canonical base pair between two identical purines. So when you have A and A, then it is called a homopurine interaction. You can also have a A and C, this is purine and pyrimidine but non-canonical. Okay, you can have a C and C, which is again non-canonical, but it is between two pyrimidines which are identical. So C and C, this is a homopyrimidinic. So you can have in all cases homopurinic or homopyrimidinic non-canonical base pairing. And of course, we already know that you can have non-canonical base pairing between G and C itself, but with two hydrogen bonds, or between U and A itself, but with one hydrogen bond. So it can deviate from the normal, and whenever it deviates from the normal, we understand it to be non-canonical. And these are all possibilities. So these have been observed in various RNA structures. Okay, this is something that has been studied through various techniques, various X-ray crystallography studies or NMR studies, or and so something called selects. Okay, so they have been able to see such different non-canonical base pairing happening, especially in the RNA molecule. Interestingly, what has been observed is just as you observe with the canonical Watson and Crick base pairing, you can also observe the same kind of intra-base spatial parameters with the non-canonical Watson and Crick base pairing as well. So you would have non-canonical Watson and Crick base pairing buckling or opening or having a propeller twist or stagger or shear or stretch. These are certain things that you observe when you look at canonical Watson and Crick, but you can also see that in non-canonical Watson and Crick base pairing, and therefore it is understood that even if you have non-canonical Watson and Crick base pairing, they would contribute to a dynamic structural change that can be seen with respect to what its surrounding is, and also what its adjacent base pair is, or it would change, or it would vary. The intraspatial base pair spatial parameter may change with respect to the moisture content. Okay, so all these can happen also with non-canonical Watson and Crick base pairing. and therefore this has an implication on various molecular mechanisms, especially related to RNA, whether it is splicing or translation or transcription. Everything is compounded by these intra-base pair spatial parameters, or affected by the intra-base pair spatial parameters within the non-canonical Watson and Crick base pairing. So here what you look at is that you have, or the understanding is that when you have a canonical Watson and Crick, you know that the N3 C6 axis is going to bind to the 2 3 4 of the pyrimidine, right? So when you have a 180 degrees turn, so this O from this position comes here and this goes over here, okay, you would still have the hydrogen bonding happening, but there is a change. The change is that the ribose, which is present as cis over here, right, has now become trans. So when you have the sugar present in a normal Watson and Crick base pairing, then that is what is called as reversed Watson and Crick. So the sugar being in the trans form, you see what is called as the reversed Watson and Crick. You will never find a reverse Watson and Crick in an anti-parallel DNA, but you can find it in a parallel DNA. So when you have two single strands of DNA coming together in such a way that you have both the strands parallel, that is one strand 5 prime to 3 prime, the other strand also is 5 prime to 3 prime. When you understand anti-parallel, it is always one strand is 5 prime to 3 prime and the other strand is 3 prime to 5 prime. So when you have a 5 prime to 3 prime and a 3 prime to 5 prime, you will always find the Watson and Crick base pair in the cis form. But if you have a parallel base pairing happening, then you can have the Watson and Crick as the reverse Watson and Crick, where the sugars are in the trans form. And you can see that it's just a 180-degree rotation of one of the nucleobases. Now, when you have so we're looking at types of, non-canonical base pairing. So reverse Watson and Crick becomes non-canonical base pairing. Then when you have the C697 phase of purine, hydrogen bonding with the N3C4, that is the Watson and Crick phase of the pyrimidine, then you have what is called as Hookstein base pairing. That was actually discovered by Hookstein when he was studying RNA with respect to a certain molecular mechanism, and he found that there is a possibility of the C697 phase, which is not the normal Watson and Crick phase, interacting with the Watson and Crick phase of the pyrimidine, thereby having what is called as Hookstein base pairing. So you can see that it is still AT base pair, but it is not the normal Watson and Crick. It is Hookstein base pairing, and you can also see that the sugars are on the trans form. Now you can have this rotated by 180 degrees. Again, if it is rotated by 180 degrees, it will still form the AT base pair, but this time because of the rotation of one of the nucleobases, you have what is called as reversed Hookstein. So therefore, both Hookstein and reversed Hookstein are again non-canonical base pairs. Wobble's non-canonical base pairing is something that all of you are very familiar with, because we know that tRNA, okay, has or shows what is called as Wobble's hypothesis, and in that we know that the 5 prime end of the anticodon, the nucleotide at that is able to interact with the codon in the mRNA in such a way that that nucleotide can actually form hydrogen bonds with not just its complementary but also with other non-complementary nucleotides. So when you look at over here, G should ideally bind to C, but it is binding to T. So therefore, the G binding is wobbling, where complementarily it should bind to C, it is binding to T. So that also is a non-canonical base pair, and such base pairing is what is called as the Wobble's base pair. So this is very commonly observed, as we say, during translation, and therefore with that phenomenon, there is one aspect that comes up is that one tRNA can bind to more than one codon. So that is very, very important. So instead of having 60 tRNA, you can do with 24, 22, 25 tRNA, as one tRNA can definitely bind to more than one codon. Now let's look at the secondary structures that are formed by RNA, secondary and tertiary structures that are formed by RNA. And in many of these interactions or formation of the secondary structure, the complementarity that is formed, or the secondary structure or the tertiary structure that has been formed, is not because of Watson and Crick base pairing, but it is because of non-canonical base pairing. So one of the most common non-canonical base pairing that leads to a secondary structure formation is a stem structure. Then of course another common feature is the stem and the loop structure. So this is more commonly called as the hairpin loop. So here you may have again non-canonical base pairing. These do not base pair and loop out. So this is something that is a very common secondary structure in RNA. Interestingly, when you have a secondary structure, or in fact this is more found in the tertiary structure, but when you have two stems, so this is one stem, this is the second stem, this is one loop and this is the other loop. So when you have two stem, two loop structure formed, then that is what is called as a pseudoknot. Okay, when you have a single bulge, so therefore this nucleotide is not able to interact at all with this, okay, then it forms an RNA bulge. You can have an internal loop, okay, so you have this stem, you have this stem, and you have a loop in between, so that is what is called as an internal loop. You will find this present in many RNA. So when you have, for example, a bridge formed between three stems, this is the only part which is not forming a base pairing, then this is what is called as a multi-loop structure. Now let us look at how all of these things can be present in one RNA. So when you see that one RNA, one new polynucleotide RNA folds or forms the secondary structure, you can find that it forms the stem structure, it forms the bulge structure, it forms the interior loop structure, it forms the hairpin loop structure, it forms a pseudoknot structure, it forms a multi-loop structure. Everything is possible within one single RNA. So you can very clearly observe how a primary structure of RNA folds into a secondary structure and can also go into a tertiary structure. So these are all motifs that are present on folding. Okay, and the folding is not just because of Watson and Crick base pairing, but it is because of non-Watson and Crick base pairing as well. So this is the perfect example to show. This is the tRNA. The tRNA has a cloverleaf structure. So as you can see, this is the acceptor arm where you have the CCA at one end. You have the dihydrouridine arm, the pseudouridine arm, and you have the anticodon arm over here. When this loop, hairpin loop, interacts with this hairpin loop, okay, you can see how this hairpin loop, okay, interacts with this. Again, you can have non-canonical base pairs, or you can have just simply van der Waals, hydrogen bonding, hydrophobic interaction, ionic interactions, etc. possible. Now from this secondary structure, it is forming a tertiary structure. So this inverted L that you can see has been aided because of such base pairing. This is one example of how pseudoknots are formed. And pseudoknots are typical structures that are present in many RNA viruses. And also what is notable is that many pseudoknots have catalytic activity. So for example, ribozymes that are there, you would find many of the ribozymes having these pseudoknots, and these pseudoknots act or have the catalytic activity. So for example, self-splicing introns. The self-splicing introns would have a pseudoknot region, and that pseudoknot region is responsible, say, for the actual splicing mechanism. The transesterification reaction that happens is because of the pseudoknot formation. So you can see how by different ways you can have a primary RNA molecule getting converted into secondary and the tertiary structure. So you can see over here a typical hairpin loop structure being formed. Then you can also have the pseudoknot structure being formed. This is the SARS-CoV coronavirus, which you can see once it folds, you can have two stems and two loops at least present. So you have a pseudoknot structure definitely present. But other than that, you have the stem one. So here you have just this loop formation with a loop over here. So this is not typically a hairpin loop, but you can have the stem structure formed as well. So this is just an example of how you can have the primary structure of RNA folding to come to a tertiary structure. So let us look at just instances of how non-canonical base pairs have a role to play. So when you have a GU base pair, okay, this was in fact one of the first non-Watson and Crick base pairs that was observed, and it was proposed by Crick, okay, and because of this non-Watson and Crick base pairing, it could account for the degeneracy of the genetic code. The degeneracy of the genetic code suggests that um, you can have more than one codon for an amino acid. That means if you have, say for example, lysine, or say for example you have serine, then you can have 4 or 6 codons present for serine. So that degeneracy was addressed by the fact that you can have between the codon and the anticodon not Watson and Crick base pairing. This is what is understood by Wobble base pairing. A curved RNA helix. So when they studied the RNA helix, and when the RNA helix was found to be curved, it was observed that the curved RNA helix could incorporate an internal loop with GA and AA. You can see both of these are non-canonical Watson and Crick base pairing. So an internal loop within an RNA helix is formed because of non-canonical base pair. We all are familiar with signal recognition particle. Every eukaryotic cell, when you look at endomembrane network and when you look at molecules or proteins that are either secreted or they are moving to the plasma membrane, or they are sorted and transported to different places in the cell, then the first step to take the mRNA to the endoplasmic reticulum is through a molecule or through a ribonucleoprotein, which is called as signal recognition particle. This is not typically a protein, only a protein. It is basically a complex of protein and RNA. And the study basically showed by Dudna et al. That, and she's the one who won the Nobel Prize recently. The protein RNA complex that is formed, the RNA within its structure has very specific bulge or a very specific internal loop that is responsible for the protein of the signal recognition particle to bind to the RNA. So a distorted RNA helix is the site of protein binding. So there are many studies that have suggested that these different non-canonical base pairs, which lead to different secondary or tertiary structures of the RNA, those sites are the regions where the proteins can actually come and bind to. Okay. Now the other important facet is that the cellular functions of most RNA molecules, whether it is protein binding or whether it is non-Watson and Crick base pairs, like I mentioned, they are the sites where the proteins come and bind. So the RNA helices seem to have a deeper groove and more distorted helices, and therefore these deeper grooves are sites where the proteins can come and interact with. Another important fact is that a short oligonucleotide can come and bind to a double helix to form a short triple helix structure. This triple helix structure is also because of non-canonical base pairing. So these are certain aspects that one observes. These are just one few examples, but it is unimaginable when you look at all the different molecular mechanisms, whether you look at translation, whether you look at splicing, all these things you would find n number of different non-canonical base pairing involved. Okay, the stem in the acceptor arm of the tRNA itself has non-canonical base pairs, and those non-canonical base pairs actually help in the process of translation. The interaction with the mRNA at the decoding center between the anticodon and the codon itself is an example of how the non-canonical base pairing is helping in a molecular mechanism. So therefore, let us come to the conclusions. A minimum of 2 hydrogen bonds can be formed between nucleobases to form a non-Watson and Crick base pairing. And because of those hydrogen bonding between non-canonical base pairs, there are 12 families of base pairs. Every nucleobase, purine or pyrimidine, has 3 edges. One is what is very conventional, and that is the Watson and Crick edge. You have the Hoogsteen edge and you have the sugar edges that can form hydrogen bonds with an opposite nucleobase. The sugar conformations, the tautomerism, and the isostericity are factors that can influence the non-canonical base pairing. The non-canonical base pairing are most frequently observed in the secondary and tertiary structures of RNA, especially non-coding RNA with assigned biological function. The best example being anticodon-codon interaction. Therefore, there are several non-Watson and Crick base pairs that are part of DNA and RNA, which although not as stable as Watson and Crick base pairs, contribute to specific functionalities and also give structural flexibility, which is needed in various molecular mechanistics. Thank you.